Today we're talking about plant, okay? Because a lot of students, they forget about the plant. Yeah, plant also is very important in our exam, right? Okay, before that, yeah, let's start by discussing the recent past year, okay? This one. And then I already write the answer, okay, but I will give you few questions now, and then you can try to answer for it. Okay, I hope it will help you. For me for one. Okay, so please open this note. Yeah. So now we are going to discuss about this one, 2025, which is last year questions. So this show about what? What is this? It's a leaf, right? And then what is the T referred to? What is the structure of the T? It is a hydrophyte or xerophyte. What do you think? What do you think about the T? T is referred to the big holes, okay, which is between the cell. So actually this T, right? Do you know the name of T? It's known as the airspace, okay? T is known as airspace. Yeah, so that's why this is found in the plant, especially in the hydrophyte, okay? Because hydrophyte is a plant, and then this plant, right, is living inside the water. So that's why they require a lot of airspace. Okay, so that's why draw a circle around one bacillus bundle. Okay, and that's now where is the bacillus bundle? Can you see the bacillus bundle here from this picture? Can you see where is it? Can you see the xylem and phloem?
Yeah.
Yeah, so where is it? It is at here.
Yeah.
Here. Yes, here and here. And then the big hole will be the xylem, okay? The big hole is xylem here, okay? And then the smaller hole is phloem. Yeah. So just draw a circle around one bacillus bundle, so you can just draw one. And then draw a label line and the letter T to identify the palisade mesophyll tissue. So where is the location for the palisade mesophyll tissue? Where is it? So palisade mesophyll tissue, right? It is, if you recall back, palisade mesophyll tissue is usually just located under the upper epidermis. Okay, just the upper epidermis, just beneath the upper epidermis of the plant. So that's why, because palisade mesophyll cell consists of what kind of structures? So they can do the photosynthesis. So they consist of a lot of the chloroplast, right? So that's why it's located here. So draw a label line, okay, and the letter X. So you can draw like this, okay? Just use your ruler to draw a line around here. Okay, so here is a palisade mesophyll cell or mesophyll tissue. And then state the name of the cell structures in the palisade mesophyll cell. So inside the palisade mesophyll cell, right, it consists of a lot of chloroplasts. You know there's only 3 structures in the leaf, right? Because usually the leaf consists of the chloroplast, so only the leaf they can do the photosynthesis, right? And then you know what is the name of structures in the leaf which consists of the chloroplast. There will be 3 structures. One will be the palisade mesophyll cell. Another one will be which one? Which structures? Palisade mesophyll cell and also the spongy mesophyll cell. Another one is the gut cell. So there's 3 cells. These 3 cells consist of this chloroplast. And then tell me which cell, okay? Because you have palisade mesophyll cell and then you have this one, the spongy mesophyll cell, right? Spongy mesophyll cell, and then another one is a gut cell. Gut cell. Those 3 cells consist of the chloroplast. And then you tell me which structures consist the highest amount of chloroplast. Is palisade or spongy or gut cell? What do you think? Highest number of chloroplasts? Your answer is? Which structure consists the greatest number of chloroplasts? Is in the palisade mesophyll cell or spongy or gut cell? What do you think?
Palisade mesophyll cell.
Sorry?
The palisade mesophyll cell.
Yeah, okay. Yeah, it's palisade mesophyll cell. Okay. And then why palisade mesophyll cell consists a largest amount of chloroplasts is because due to their structure. And then you should know how to describe the structures for the placid muscle cell. Okay, which is the tall columnar shape, and then inside they have a lot of chloroplasts. Yeah, due to their structures. And then for the spongy muscle cell, right, the structure is they are more loosely arranged. Okay, loosely arranged. It's not as tightly packed like in the placid muscle cell. So that's why placid muscle cell they consist a lot of chloroplasts compared to spongy and the gut cell. Yeah, that's it. And then describe the function of tissue in the vessel bundle. Okay, so this is very important. I always repeat every year. It's not every year, okay? It's a common question. Yeah, this one. Function of phloem and xylem. So you should know that phloem, write about this keyword. See this keyword, translocations. Write about the translocation of the sucrose and the amino acid. And then xylem, okay, is involved with transpirations, right? Transpirations is help in the transport of water and the mineral ions. Another thing is xylem will help to provide the support, the mechanism support. It's because xylem consists of the lignin. They consist of lignin, so that's why they can help to provide support. Okay, and then this is a plant live in the water, so that's why this the plant known as hydrophyte, and then hydrophyte is live in the water, so that's why what kind of features to allow the plants to float on the surface of water. So this is airspace. So you can see right, this small hole, right, it's not small hole, it's a big hole. So those are the airspace. There's a lot of airspace between them. So the airspace, right, they will provide the buoyancy. Yeah, the function is they will help to provide the buoyancy to the leaf so they can float on the surface of the water. Okay, provide the buoyancy. I want to see this keyword. You can say buoyancy, or you can say that help to reduce density, which is the leaf will become lighter so they can float on the water surface. And then, okay, explain other adaptations of this group of the aquatic plant. Okay, this hydrophyte consists of what kind of adaptations other than they have this airspace? What do you think? Oh, so you can say that there's a stomata. So because of the plant living in the water is different from the plant living on the land. Previously, we usually study about the terrestrial plant. Okay, the terrestrial plant is referred to the plant just live on the land. So that's why for these terrestrial plants, right, the stomata usually located on the lower epidermis of the plants. Why the stomata will located at the lower epidermis of plant, which is in the normal plant that they learned before, right? Which is this one. Okay, this one. This chapter 6, right? Which is here. I think it's here. The structures, okay? I didn't state it here. I think it's here. The structure of the plant. Okay. Do I put the picture here? Oh, I didn't put it. Okay, so just you need to know about the leaf structures, right? The leaf structure. Because usually what you learn before is the leaf structure. Okay, the upper part will be the upper epidermis, right? And then here will be the tall columnar placid muscle cell, right? And then here will be a lot of the spongy muscle cell, yeah. And then followed by this one, and then there will be the lower epidermis. Okay, here is lower epidermis. So between the lower epidermis, there will be the gut cell. Okay, around here is the gut cell. Yeah, gut cell. So that's why here is the stomata. So in the terrestrial plants, which is live on the land, the stomata is located at the lower epidermis, not really at the upper epidermis. Okay, so why they need to locate it at the lower epidermis? What is the reason? It's because when the stomata they located on the lower epidermis, they can help to reduce water loss. Okay, they can help to prevent the water loss. Because if the stomata located on the upper epidermis, right? So usually the upper epidermis is directly exposed to the sunlight. Yeah, because when the sunlight they strike on the plants, right? They just shine on the leaf surface. So that's why if the stomata located on the upper epidermis, there should be a lot of water loss during in the evaporations of the water. okay, during the evaporation of water. So that's why, but this is different in the hydrophyte plant, okay, which is in the aquatic plants. So why does stomata need to locate at the upper epidermis? Okay, it's because that the aquatic plant, right, they are living in the water. So that's why the plant need to float on the water surface. So that's why their lower epidermis usually in contact with the water. So that's why they want to prevent the stomata located on the lower epidermis. Yeah, so that's why stomata usually located on the upper epidermis. So that's why they can allow for gas exchange in the air. Okay, so this is one of the feature. Stomata at the upper epidermis. Another one is the hydrophyte. They have thin or no waxy cuticle because they are live in the water. Okay, thin and no waxy cuticle. Not like in the terrestrial plant. In the terrestrial plant, we have a waxy cuticle, much more thicker than the aquatic plant. Why the terrestrial plant, they should have the waxy cuticle? Because they want to, as a waterproof layer, okay, to prevent any water loss. Because they want to preserve the water. So that's the difference, okay? Hydrophyte, no need to scare about water loss. Because hydrophyte, they just live in the water. Okay, so that's why they just already live in the water. So there's no need to prevent the water loss. So that's why they should have a very thin and no waxy cuticle. 2 marks, so that's why this is the... Okay, whenever they ask about this one, explain one other adaptations of this group of aquatic plant, right? Okay, after you mention the adaptations, adaptation, right, is this one, okay? Stomata on the upper epidermis, thin and waxy cuticle. And then, but you should elaborate the point, okay? Why you should have stomata and upper epidermis? Because you want to allow for gas exchange. So this is the way you need to answer this question. Explain the reason why you should have this kind of adaptations. Okay, you have a thin, no waxy cuticle because your plant doesn't need to preserve any water. They are not required to prevent water loss. So explain the reason behind. Okay, and then what means the adaptive feature? Adaptive feature, okay, this is actually in the variation. I think it's variation chapter. So it's referred to the inherited feature. They help the organism to survive and reproduce in these environments. And then here is the grass leaf. See? The grass leaf, okay? And then this is what? This is the hydrophyte or xerophyte. Okay, what do you think? What is this? This is what type of plant? What kind of plant is it? Is it a hydrophyte or xerophyte? It is a xerophyte, right? It's a xerophyte, okay? How you're able to recognize it is a xerophyte? You know, xerophyte is referred to the plant which is live in the dry, hot environments like the cactus. Yeah, so it is a xerophyte. Okay, let me show you the how does it look like. Okay, so we can refer to this chapter 18 now. Okay, because this two part, right, which is we learn about the hydrophyte and xerophyte. This is the contents inside the chapter 18 now. Okay, so we can read back. See this one, adaptive feature. Yeah. Okay. And then, okay, this one, how are hydrophyte adapted to the environments? So that's why this is the adaptations. Yeah, you can memorize it if you want. So hydrophyte, right, usually it's just like this, right? Okay, their structure is they have a very thin cuticle on the upper epidermis. And then there is a stoma. Actually, there's stoma. Stomata located on the upper epidermis. And then the placenta mesophil cell. So you can see right here is the placenta mesophil cell. Yeah, so that's why you should maybe just like the exam asking, right? So yeah, wait for a while. Okay. Yeah, so the placenta mesophil cell, right, is located below. You can see, right, just below the upper epidermis. Just below the upper epidermis. So that's why they can help to maximize the photosynthesis. And then there's the airspace. So you can see, right, the airspace here. So they can provide the buoyancy. And then below that would be the lower epidermis and also covered by a thin cuticle. So just remember it's thin cuticle. So here's a point, okay? They have a large airspace to provide them with buoyancy. They have a small root. Okay, usually in the hydrophyte, right, they have a small root. Why? It's because they can just extract nutrients directly from the surrounding water. So they are not need to require have such a long roots to penetrate far to get the water or nutrient. No require. So usually their root is very small. Stomata on the upper epidermis. So that's why they can allow the gas exchange easily with the air in the atmosphere. And then there's lack of stomata at the lower epidermis due to the underwater, okay? Because if your stomata located more on the lower epidermis, right? It's just like your nose, okay? Your nose is being immersed in the water. You cannot breathe. Okay, airspace for buoyancy. Okay, airspace not only for buoyancy, right? But airspace also they have second function, which is they can allow for the diffusion of gas and the water vapors. Okay, they can allow in the exchange of the gas, which is they can allow the carbon dioxide to diffuse inside and the oxygen to be released out. And also they can help to release in the water vapors. So they are not required to conserve water, so that's why they have lesser or no wax cuticle. Okay, that's all. So be clear about hydrophyte, because hydrophyte, right, is a hot question now. Okay, I think almost every year they will ask at least 1 or 2 questions about this hydrophyte. So you just remember about the structures. And then second one is xerophyte. So this one is xerophyte. Okay, so how are xerophyte adapted to the environments? So xerophyte, right, is like this. Okay, you should know about the features. So this is the hair. Oh, this is the hair, okay? The hair structures. And then another one is different from the hydrophyte just now. Xerophyte, right, they have a very thick waxy cuticle, okay? So this outer surface layer is referred to the upper epidermis. And then the inner surface area, they are referring to the lower epidermis. And then where is the stomata? Can you see the stomata? Can you see the stomata in the xerophyte? It's not too obvious, right? But here's a location for stomata. So you can see, right? You call it what? Sunken stomata. This is a special feature only found in the xerophyte. Sunken stomata. Just like this, okay? See? The stomata, right? It just like have an inner groove inside. So that's why they can help to trap more air. Also, sorry, no. They can help to trap the water vapor or the moisture. Okay, so generally, just like this, okay? And then here is a structures for the xerophyte. So xerophyte, they have a very thick waxy cuticle because they want to prevent water loss. Then they have a sunken stomata. So you can see right here stomata. So stomata usually found in the pits to trap the moist air. So this is their purpose. They want to trap the moist air and then to allow to build up the humidity layer inside. So they can help to reduce the water loss by evaporation. Because xerophyte, right, they are just trying their best to conserve more water. Because they really require a lot of water, okay? They try their best to get more water. So they use this sunken stomata help to trap any moist air inside. So as they can reduce the rate of the evaporations of water to prevent further water loss. So this is their purpose. And then this xerophyte, right, they have a roll leaf. Roll leaf just look like the leaf, look like needle, or sometimes you call it needle-like leaf shape. Yeah. So this roll leaf, right? Roll leaf, okay, they can help to trap the moist air and prevent the air movements across water to reduce the water loss by transpiration. Just like the sunken stomata, okay, just want to trap the moist air. They want to conserve the water vapor inside. And then they have a small needle-shaped leaf. This is help to reduce the surface area for transpirations. Okay, so that's why they can losing small amount of water because the surface area is very small. And then they have a hair inside, okay, this hair, see, like this is hair inside. So hair also help to trap the layer of moist air. So the keyword, trap layer of moist air, reducing transpirations, okay. So this is the keyword, okay, trap moist air. And then they have essentially shallow roots, allow for quick absorptions of large quantity of water when it rains. So actually, right, at the Suya, 05, they have shallow roots. Okay, they have two types of root system, you know. One is shallow roots, one is because deep roots. Okay, because usually, right, in the desert, I mean the climate is very hot. And then whenever there's raining, okay, so the plants will try their best to absorb the water from the soil surface after raining. So that's why they have essentially shallow roots. But they also have a deep root as well. Okay, so this is very important. You should know about structure in the hydrophyte and xerophyte. Okay, and then describe the physical feature that identify this as a xerophyte leaf. Okay, you can see what kind of feature. You see the hair inside, right? See the hair? All right, you can see the hairs. And then you can see the stomata. See? Around here, stomata, here. Here. Here is the location of stomata. It's not very obvious, but this around here, which is found inside the pits. You can say stomata are found in the pits. And then they are called Sanger stomata. Okay, hairs inside. And then they have very thick cuticle. Thick cuticle on the upper epidermis. So these are features, okay? Curl and roll leaf to reduce the transpiration, to reduce the water loss. And then this is just simple calculations. Just change the micrometer to millimeter. And then this one, carbohydrate molecule used to build a cell wall in the xerophyte. Okay, just remember that whenever the cell wall is in the plant, the material are always below the cell walls. Then how about the cell wall in the bacteria? Is made from what kind of component? And also, so remember, the component of cell wall in the bacteria, what's the name of the structures? Start with P. Yeah, peptidoglycan, well done. Okay, how about the fungus? How about fungus? Cell wall in the fungus, made from what kind of structures? Start with C. So remember, cell wall components in the fungi. Chitin. Chitin, this one. Okay, this chitin is a strong structure. Okay, this chitin also found in the exoskeletons in the insect. Yeah, they also become part of the exoskeleton in the insect. And then this one, okay, suggests one way the stem of xerophyte is adapted from the dry habitats. So how they can adapt for this kind of dry habitat. So that's why they have a swollen or succulent stem, which is their stem is very, very big, very, very swollen. So inside they can store a lot of water for water storage. So you can see, and also there's three types of root system in the xerophyte. We call it what? Extensive shallow root system or the deep root system. Okay, so that's why you should know to explain the reason. Yeah, so species 1, they has a shallow roots. They can spread out to absorb the water after the rainfall from the soil surface. The species 2, they have deep roots. Okay, they have deep roots to absorb the underground water. You know underground water. Actually, there's water in the underground. Yeah, the groundwater. Groundwater will cause what? 地下水. Okay, yeah, 地下水, which is found deep in the soil. So they need to penetrate. I mean, you can see their roots, right? They will just grow longer and longer, trying to search for the water source. Okay. Okay, again, repeat again. 2025. I think last year they come out. Last year they come out a lot of these kind of questions. Yeah, hydrophyte again. Okay, ask you to describe the structures of the hydrophyte. Then, okay, what's A? A is referred to what structures? A? B? What do you think? I'm waiting for your answer. A is referred to what kind of structure? A.
The upper epidermis.
Yes, it's upper epidermis. How about C? Lower
epidermis.
Yes, well done. Okay. How about B?
Spongy mesophyll.
Spongy mesophyll. Okay. Pellucid mesophyll. B is placid mesophil cell. It's not spongy mesophil cell. Okay, where is location of spongy mesophil cell? Spongy mesophil cell, right? They are around here. Just remember that placid mesophil cell, right, is just located under the upper epidermis. So you can see here is a part of the cell, right? This part of the cell they just found under the upper epidermis. So that's why this is belong to placid mesophil cell. Then the spongy mesophil cell, right, is usually closely associated with the airspace. So here is the starting from this layer. Okay, starting from this layer will be the spongy mesophil cell. Yeah, which is here. See here. They are all belong to a spongy mesophil cell because they have they are closely associated with the airspace. See, here is the airspace. So this is just same in the terrestrial plant, right? In the terrestrial plant, the spongy mesophil cell is just very, very loosely arranged, and then there's a lot of airspace between them. Okay, so C is epidermis. Or you can say lower epidermis. It's up to you. Just I want to see this keyword. Okay, epidermis. Okay, two functions of the water lily area labeled D. So D is what? D is the airspace. Okay. Oh, D the airspace. So that's why you need the airspace to do what? You need airspace for two reasons. To provide the buoyancy, and then to allow for the diffusion. Okay, diffusion of gas, buoyancy. Okay, diffusion of gas, right? Allow for diffusion of the gas, which is for the carbon dioxide and the oxygens, and also they can allow for the diffusion of the water vapors during transpirations. Okay, you should mention the keyword diffusions, water vapors, or the gas. Okay, and also the name of process. So photosynthesis and the transpiration. Because transpiration, right? Transpiration, so remember the definitions. If you refer to evaporation of the water through the stomata. Yeah, evaporation of water through the stomata from the airspace. Okay, and then green pigment found in the tissue B, right? So that's why the green pigment is referred to the chlorophyll, because only the chlorophyll they are known as green pigment. So what's function of the chlorophyll? Yeah, they do ask about function of chlorophyll. So you should say that chlorophyll is the structures they can help to trap light energy, and then they help to transfer the energy. Okay, the keyword trap light energy and then convert the light energy become chemical energy to produce the carbohydrates. Yeah, so you can see my answers like step by step. Okay, use light energy to convert from the chemicals. Oh, sorry, from the chemicals to produce the carbohydrate, to produce the glucose. Okay, yeah, here's the adaptation. See, repeat again. So just copy the point just now. Okay, I should write the thin critical. Okay, and then stomata on the upper surface. Yeah, and then because you want to explain, right? So usually you just write about one adaptation. Means that you're writing about one feature, and then you need to describe why is it, why you need like thin critical. Okay, you need thin critical because you want to allow more light penetrates to the placid cell layer to do the photosynthesis. You need the stomata on the upper surface because the lower surface is on the surface of water, okay, or under the water. So that's why stomata only can be at upper surface side. You want to prevent them to okay, I mean you want to prevent the stomata to in contact with water, right? So explain it. Okay, this is very important. Explain it. And then this one, stomata density and habitat of two different hydrophytes. So there's two type of plant, pondweed and water lily. So these two plants, right, they are also living in the water. And then why pondweed don't have stomata? See, there's no stomata on the upper leaf and also no stomata on the lower leaf. And then for the water lily, right, yeah, they have more stomata on the upper leaf. The lower leaf, right, they don't have any kind of stomata. So they why, okay, convert the stomata density of the upper leaf surface of the water lily to the cm cube. Okay, this is just calculation. Just calculation. Okay, this is more challenging. So we can see, right, explain the stomata density found in two different hydrophytes. Okay, why pondweed? Totally no found any stomata at the upper leaf and lower leaf. So that's why, because hornweave, right, is submerged plant. Hornweave, okay, the difference between this submerged plant is referring to the submerged plant, right, is just the plant totally immerse themselves in the water, totally submerged in the water. Okay, not like the water lily. Water lily is a floating plant. Okay, most of the hydrophyte that we learn is floating plants. So that's why they have no airspace to allow them to float on the water surface. Okay, so in the submerged plants, there's no stomata, okay, no requirement stomata, because the oxygen and the carbon dioxide, they can just diffuse directly from the leaf, to the leaf from the water. Because stomata is intake the gas by the gaseous state, right, which is the oxygen and the carbon dioxide, they just go in and out of stomata through the gas form. Since this submerged plant, right, already fully immersed in the water, so there's no, they cannot contact to the air. There's no air contact to them, right? Only the water. So that's why they are not requiring stomata, because the oxygen and carbon dioxide, they can just directly diffuse into their leaf cell, okay, to the leaf cell from the water. So no need to go through the stomata. So which is in the liquid form, okay? Which is in the liquid form. Yeah. Because in the water, right, you have dissolved oxygen and the carbon dioxide. So that's why they can just directly into the leaf, which is in the water, okay, from the water. But for the water lily, okay, they has a stomata only on the upper surface. Because of what? Because the lower surface is under the water. Okay, so that's why there's a constant water supply, so there's no risk of wilting. Okay, and then and then in this lesson, I think 3 years, starting from 2022 to 2025, right, they come out a lot of this kind of picture. Yeah. So that's why you see the picture, the actual picture are like this. Okay. So that's why you need to know, recognize the picture, okay, what the exam asks about. What the question asks about this. Yeah, so this is showing a farmer. So what is farmer doing? Farmer, right, they are planting their monocultivated crop plants in the waterlogged soil. See, waterlogged soil, okay? Yeah. So identify the characteristic. They can be used to classify rice and monocultivated plants. Yeah, they should have the parallel vein, okay? And then you can say that they have what? Parallel vein also, and then they have the how many flower pattern inside. Okay, so you need to recall back the feature. And then this one, okay. So waterlogged soil contain a very low concentration of the oxygens. Right. So whenever the roots is immersed in this waterlogged soil, can the root breathe? Okay. The roots, right, inside they consist of the root hair cell. Okay, root hair cell, right? And then root hair cell, they need to do what? What is the function of root hair cell? The function of root hair cell is they will absorb the water and the mineral ions. Water by osmosis, mineral ion by the additive transport. And then since the additive transport require what? Additive transport require the energy supply to them. Okay? Where you get the energy from? Energy from the aerobic respirations, okay, generated from the respirations. But so that's why, since the energy generated from the respiration, from the aerobic respiration, right? So means that you need the constant supply of oxygen to the root hair cell, so they can use the oxygen to do the respiration, so they can release more energy to the root hair cell. So that's why they have more energy to transport the iron. To actively transport the iron. But the problem is you have a lesser amount of oxygen. Okay, so that's why in the waterlogged soil, you should say that lesser amount of oxygen. So that's why you need to the reduce. Okay, reduce aerobic respiration occurs. So that's why you have less aerobic respiration, right? So that's why you release less energy. Okay, so less energy required for the additive transport of nitrates. Yeah, and then still remember, you need nitrate ion to do what? Nitrate ion is very important, right? Because nitrate ion is the ingredient to make out the proteins. Nitrate ion is a source of nitrogens. Okay, this is an important keyword that you should remember. Nitrate ions is used up by our animal cell and the plant cell as a source of nitrogens. Why? Because you require the nitrogen elements to help to build the amino acids. So this amino acid will help to build the proteins. So if you have lesser nitric ions, why you have lesser nitric ion get into the red hair cell is because you have less energy for the energy transport. Because nitric ion only can go into the red hair cell by the active transport. So that's why you have lesser nitric ion, you have a lesser source of nitrogens, means that you have a lesser amino acid being produced. So means that you have a lesser protein synthesis. Okay, so yeah, you can write in your own answer lah. But here's a keyword. Okay, yeah, this one, less protein synthesis. And then what will happen if you reduce the aerobic respiration, right? And then yeah, in this condition, right, there might be more denitrifying bacteria presence. Okay, and then they may also be less nitrogen fixing bacteria. Okay, so this will result in the fewer nitric ions in the soil available for the plant. Okay, because you have less nitrogen fixing bacteria. You know what nitrogen fixing bacteria do? Nitrogen fixing bacteria, they will convert the ammonium compound, the ammonium ions, right, become what? Become the nitrite, and then later become nitrates. And then what denitrifying bacteria do? Denitrifying bacteria, right, they will help to convert yeah, which is this one, okay? And also nitrogen cycle is very important lah. Where is it? Yeah, this one. Nitrogen cycle. Okay. Here. Okay, so this nitrogen cycle is also very important. It's a common question, okay? So usually they will ask this nitrogen cycle together with the protein synthesis, because nitrogen cycle, right, involved in what? Involved in the nitrates. So you should know about the process, okay? The whole process, which is the three process from the nitrogen fixing bacteria, okay? Then to the nitrifications, then to the denitrifying bacteria. So you should know these three processes very well. Okay, and then just we talk about denitrification, right? Why we talk about denitrifications? Because you have the nitric ions. Okay, you have a nitric ion, even though your nitric ion is lesser, but these nitric ions, they can do what? They can undergo the denitrifications by the denitrifying bacteria. So this denitrifying bacteria help to break down the nitric ions. So nitrate, which is NO3 minus, they can become the nitrogen gas again. So nitrogen gas, right, will be returned to the atmosphere. Okay, so not only denitrifying bacteria, another source will be the lightning. Okay, lightning, they can help to produce the they can help to they can help to carry out those processes also. Okay, so nitrogen cycle. And then for this one, I think I can show one questions. Where is the questions? Let me show you the questions. I forget where do I put the questions. Chapter 8. No, I think it's at here. Yeah, let me show the question. Chapter 8. Okay, so remember. Oh, sorry. Remember the leaf structures, right? The leaf structure, remember, okay? Upper epidermis, the plastic medulla cell, and the spongy medulla cell. Yeah, and then you should know how to describe the leaf structures, okay? So here's a way. Yeah, so plastic medulla cell, right, is tightly, is a columnar in shape, located under upper epidermis, and then they contain many chloroplasts. Yeah, so they can maximize the amount of energy trapped by the chloroplast. And then for the spongy medulla cell, right, they are loosely packed. So that's why the spongy medulla cell, usually they are associated with a lot of airspace. So this airspace, they can help to allow the gas exchange and diffusion of gas. Okay, so they can also allow the evaporation of water. Okay, so water is being evaporated from the cell surface to the airspace. Okay, so here's it. I want to show about one question. Let me find. Where is it? The questions. It's a good question, sir. Okay, I want to show these questions. This one. Okay, 2021. Okay, let me go through with you for this question for a short while only. Actually, I will send the answer to you so you can later you can read back. So this is plant roots, right? And then because they always repeat this similar style of the questions. So see, nitrates, okay, uptake of the nitrate ions by the root hair cell. So here is the distance from the root tips, right? So see, distance from the root tip. So once the distance from the root tip become more and more longer, further, so it equals the rate of nitrate ions uptake, right? Starting from here, right, they slowly increase, and then they reduce again. And then describe the rate of uptake of nitrate ion along the roots, right? So nitrate ions, okay, initially they increase to peak and then subsequently decrease. Okay, you need to describe the overall trends. Okay, 5 marks. Okay, because they ask you to describe. Okay, describe refer to what? To describe the trend of graph, right? So initially it's increasing, right? Initially it's showing increasing trend, but later it's decreasing. So that's why you need to write this. Okay, so and then how about the number? The maximum uptake occur as 27 millimeter from the root tips, which is like here, okay, around here, maximum uptake. So which is around 27 of the distance from the root tip, right? They absorb most of the nitrate ions by the root hair cell. Okay, reaching, yeah. So and then here's the rate. Rate is referred to here, okay. Oh, sorry, which is here, 28. Okay, here 28. So that's why when you describe the graph, right, it's better for you to write about the trends, increase, decrease, plateau, yeah, and then include about the value also, okay, the figure from the x-axis and the y-axis. And then there's no there's no recorded uptake of the nitrate ions before 2.75 millimeter from the root tips. So before 2.75 millimeter from the root tip, right, which is around here, okay, 2.75. Before that, there will be no nitrate ion being absorbed by the root tips, okay. But later, the uptake levels between the 5 millimeter and the 15 millimeter from tips, yeah, which is around here, okay, from 5 to 15, yeah. So slowly there's some of the nitrate ion being absorbed, being uptake by the root hair cell. Okay, so this pattern corresponds to the root structures when there's no root hair cell found at the root cap. No root hair cell found at the root cap. Do you know what's meant by root cap? Root cap is referred to like this, okay? So this is a root tip, right? Yeah, this is a root tip. If you want to draw something like this, okay? This is the root tip. Root tip is until here only. Yeah, root tip. And then root cap is just a protective layer cover the tips of the roots. So this part, right, is a root cap. Just like you are wearing a cap on your head part. So this root cap, it just helps to protect the root tip only. So it will not help in any kind of functions. Okay, it will not help the root tip to absorb the mineral ions. No, they just want to protect the root tip only. So that's why, yeah, there will be no more nitrate ions absorptions whenever they are at the root cap regions. Okay, but slowly, okay, the nitrate ions absorptions is increasing. Okay, so and then you can record the highest uptake, which is between 20 to 35 millimeter, around here, 20 to 35 millimeter. So, which is a lot of the root hair cells are located. Okay, a lot of root hair cells are located. So you can see, right, 5 marks. Describe the graph. How you describe it? They just ask to describe, no explain. Okay, so that's why this is the answer. Do I write any reason? No, I just write about whether it's increased, whether how's the trends of the figures. Okay, I just write the figures. So that's why you don't need to explain that if the question didn't ask you to explain. So I just remember any kind of iron, no matter it's nitrate ions or other kind of iron, like sodium ion, chloride ions, whenever they just want to move into the roots part, right? They need go through the anti-transport. Okay, so here's a point for the anti-transport. Okay, write the energy against protein carrier, protein pump. Here's a point. 4 marks. Okay, this kind of question should be very easy to score. Just write the keyword on it. Okay, and then explain why uptake of iron such as nitrate is important for uptake of water in the roots. So why is very important for the plant to uptake nitrate? It's because nitrate is a source to make the protein later. Okay, and also not only this, and also the accumulation of nitrate, right? It can help to lower water potential inside the roots. Okay, so that's why inside the cell step of the root hair cell, they have a very high concentration of nitrate ion. So this high concentration of nitrate ions, right? So which is like this, just imagine this is a root hair cell. This is a root hair cell, right? This is the root cap, and then inside they have a very high. Oh, sorry, why I'm drawing sodium? I should draw the NO3 minus, which is nitrate. Yeah. So inside of this cell root hair cell, right, they have a very high concentration of the nitrate ions. Outside, they have very low concentration of the nitrate ion. So the water will diffuse inside. Okay, because outside they have a higher water potential due to the very low concentration of nitrate ions, but inside they have a lower water potential due to the high concentration of nitrate ions. So just remember to mention, okay, the osmosis, move in the roots by osmosis. Okay, so another thing is you need the nitrate because you want to do the proteins off. Okay, nitrates is used by the plant to produce the amino acids. So later amino acid, right, is an essential unit to help to build the protein later. So what kind of protein is made by the plants? For example, enzyme. Okay, enzyme, which is involved in the metabolic and structural process. And okay, this one, okay, I put a star here. So whenever the question about this one, how nitrate ions are being produced in the soil. So it means you need to include about the nitrogen cycle. Okay, so where did nitrogen ion come from? The nitrate ions come from the ammonium compound, right? And then ammonium compound, right, you get from where? You get the ammonium compound from the nitrogen fixing bacteria. Okay, because ammonium compound is initially is from the nitrogen gas, right? Which is here. Where is it? Yeah. Okay, from the nitrogen gas being converted by the nitrogen fixing bacteria. Okay, become the ammonium compound. Yeah. So later the ammonium compound, right, they will become nitrites and then nitrates. Because the plant only can absorb in form of nitrates, which is this one, NO3 minus only. Plant cannot absorb ammonium compound or cannot absorb the nitrite, which is NO2 minus. No, they only can absorb this NO3 minus only. Now, so that's why nitrate becomes available in soil whenever the organic material such as proteins are decomposed by the microorganism. So that's why this process calls about nitrifications. So the bacteria, okay, which is bacteria, they will convert the ammonium ions, the NH4 plus become the nitrite and then nitrate. So this process shows about how the bacteria produce the nitrates from the ammonium compound. So this is being carried out by the nitrifications bacteria, okay? So later, the nitrates also can be generated through the nitrogen fixations, okay, where the nitrogen from the gas is being converted to ammonium by the nitrogen fixing bacteria, and also by the natural event like lightning, okay? lightning refer to the a kind of the natural phenomena. So that's why. So here is a summary. Okay, from the amino compound become nitrite and nitrate, right? This is by the nitrifications. But for the lightning, they also can help to convert the nitrogen gas become the nitrates. Okay, which is they can help to convert this nitrogen gas from the atmosphere, because nitrogen gas, right, is in the gas form, which is present in the atmosphere. So they can convert become the ammoniums or the nitrates. Okay. So generally it's like this. Okay, just remember to mention about the process. Nitrogen fixing bacteria, nitrifications or denitrifications, or the lightning. Okay, because nitrogen cycle are also very common question coming in exam. So remember it, okay? And then I teach anyway already. Okay, which is, okay, go back to the question just now. Okay, so you know about the whole process, yeah? Okay, I hope you know about whole process. Which is this one. Why in this condition, right? More denitrifying bacteria present is because you want to convert the nutrients, okay, which is this one. You want to convert the nitrates, become the nitrogen gas, and release back into atmosphere. So that's why you need the denitrifying bacteria. And then you also have a less nitrogen fixing bacteria because you have a less amount of the nitrate ions. Okay, and then for this one, this is in the chapter 6 or chapter 8. This is talk about what? The temperature. I think this is in the chapter 6. What kind of factor will affect the rate of transpirations in the plant? So here is the temperature, right? Increasing. So you can see it's increasing. But the rate of transpiration, right? Initially they will increase, but they will reach until plateau stage. So describe the result. Describe the result, yeah? Result. Not explain, describe it. So you should say that initially the rate of respirations, or sorry, the rate of transpiration, they increase as the temperature rise, increase, and then later they will become constant and reach a plateau state. Another thing is you compare, okay, the higher rate of the higher rate of the transpirations at the lower surface compared to the rate of transpiration at the upper surface. Yeah, just compare, describe only, okay, describe. So because a lot of students, right, whenever they ask to describe, they will put the reason. So the answer only describe the trend. Unless they ask you to describe and explain the result, only you write the reason. Okay, explain the reason for the shape of the graph for the upper surface of the leaf X and Y. X and Y, okay, what is this? So so you can see, initially it's in the X state. If in the X stage, right, the temperature is actually increasing, rate also increasing. Yeah, unless they reach a Y state, right, the rate of transition just remain constant. And then Y. So as the temperature increase, they will increase the kinetic energy, okay, between the water molecule. Yeah, so that's why this will increase the rate of evaporation and diffusion of the water vapors. And then in the Y state, okay, the rate become constant. Okay, which is around this stage, see? So why later the rate become constant? Because now the stomata are fully open. And then another factor is, okay, actually there's a limiting factor take place already. So if you want to further increase the rate, you need to add on other kind of factor, which is add on the humidity, light intensity, which is now becoming limiting factors. Okay, because now your temperature, okay, your temperature is not limiting factor. So that's why you should add other conditions in order to increase the rate of transpiration. Okay, and then this one suggests how the structure of the lower surface differs from the upper surface of the leaf used in these investigations. Yeah, we also talk about the lower surface. Okay, the lower surface has more small data than upper surface. Okay, why? Because this is talk about what? They didn't show about any kind of plant, but they show about, okay, so upper surface and the lower surface leaf, right? And then this leaf is being provided with constant supply of water. Okay, so they refer to the lower surface, they have more smata, which is this one. Lower surface, right? More smata. And then this one, upper surface, right? It is more smata. Okay, so that's why overall, the rate of transpirations in the upper surface is lower than the lower surface. Okay, this show you about this one. Okay, so that's why the lower surface, right, they have more smata than the upper surface. Okay, because the rate of transpirations is higher than lower surface than in upper surface. So we can do the requires a conclusion that the lower surface lose more water, because whenever you increase the rate of transpirations, you are actually increasing the amount of water loss, right? Increasing your transpiration refer to you are actually increasing the water loss. So that's why how you're able to increase the water loss, more water loss. So actually you can allow the water being lost through the transpirations or by the evaporation of water through the smata. So that's why you have a higher rate of transpirations, means that you have a higher loss of water from the lower surface than the upper surface, right? So this means that the lower surface, they have more smata. Okay, they have a higher number of smata compared to the upper surface. And then this one, okay. So remember, okay, one thing you remember, okay, this is in the plant nutrition. So what kind of ions to make the chlorophyll? So it's magnesium. Okay, magnesium. You know, because magnesium elements, right, is found in the pigment in the chlorophyll. Yeah, in the chlorophyll, right, they have a component inside which is a magnesium. So if you supply enough magnesium to the plant, right, the plant, they can use the magnesium to produce more chlorophyll. And then this one, you want to investigate the light on the photosynthesis in the algae using the hydrogen carbonate indicator solutions. So see this again, okay? The change in color, okay, the change in color. Yeah, so that's why you can see, when the pH become more acidic, the indicator become yellow. But when the color become more alkaline, right, the indicator become purple. Okay, so two test tubes were set up in the same time, and then both contain algae and the hydrogen carbonate indicator. So at the start of these investigations, the hydrogen carbonate indicator was red in both test tubes. Red means what? Red means that they are around 8.4. Okay, 8.4. And then later they will put the test tube. One is dark, one is light. So you can have a look at. So after 20 minutes, right, the test tube in the dark will still become yellow, and then the contents of test tube in the light become purple, and then white. Explain the result. Okay, why the test tube in the dark become yellow? Yellow means what? Yellow means that the pH, right, is actually reducing. Okay, because yellow pH 7.6. And then what about the contents of test tube in the light? They turn purple. The light refer to what? The light refer to they become more in alkaline. They appear more in alkaline. So that's why they will just change purple. So we need to explain the result by showing that what happened, what kind of process happened. Is it respiration happen or is it photosynthesis happen? But we can clearly have a look at since that the test tube, right, the color of test tube change to yellow color, means that the pH, right, is more to the acidic. Okay, so that's why in the light, okay, in the light, the algae will do the photosynthesis. So when they do photosynthesis, right, they will absorb the carbon dioxide. Carbon dioxide is an acidic gas. Okay, carbon dioxide is an acidic form of gas. So this acidic form of gas, they'll be used up by the plant, right? So what will happen is now the surrounding pH, right, because they have lesser amount of carbon dioxide, so they will be increasing in the pH level, which is they become more alkaline. So that's why the indicator turn purple. Okay. But in the dark place, plant will do what? In the dark place, plant cannot do photosynthesis. Plant can only do the respirations. So when the plant do respiration, right, they will absorb the oxygens, release carbon dioxide. So they do more respirations, they can release more carbon dioxide. So carbon dioxide is an acidic gas. Okay, so just write out this one. They can do the respiration to produce more carbon dioxide. So this will cause the pH to become decreased or become more acidic. So indicator change yellow color. Okay, so just remember that the plant, okay, especially the stomata, okay, you know, stomata, they have 3 functions. They can do what? They can do the respirations, photosynthesis, and the transpiration. These 3 functions. Okay, so that's why, yeah, like this. Okay, remember the pH indicator color, okay, which is this one, hydrogen carbonate indicator. And then for this one, effect of light intensity on the rate of photosynthesis. So here's the light intensity, okay, so what will happen? So once you increase the light intensity until reach here, become plateau already. Okay, your answer is what? State the factors, okay? What kind of factor could be limiting the rate of photosynthesis at X and Y? What kind of factors? So now we talk about light intensity, right? So now light intensity, now they're not becoming the limiting factor. So the limiting factor will be other, okay? Will be others. So you can say about what? So you see at the X, right? The limiting factor is the light intensity. So that's why whenever you increase your light intensity, you actually increase the rate of photosynthesis, right? So what happens if any further increase in light intensity will not increase in the rate of photosynthesis? So they just remain constant. So that's why at the Y part, right, at the Y regions, refer to the limiting factor belong to another factor, not light intensity already. So you can write about what? You can write about the temperature or the carbon dioxide concentrations. Okay, so this one you can refer back to chapter 6. I think it's chapter 6. Yeah. Okay. So we can read through the through this document. Yeah. And then for this chapter 6, right, what's the importance point? You should know how to write the equations for the photosynthesis, and then you should know how to describe the photosynthesis process, which is it will absorb the light energy, right, by using the chloroplast. Okay, sorry, it's chlorophyll, because chlorophyll is the one who trap the light energy. So later the light energy will be converted to chemical energy stored in the glucose, and then they will use the oxygen as byproduct. Okay, so here's the equation. You need to know how to write the balanced photosynthesis equation now, okay? Because as I come out this before, remember to add the 6 in front because it's balanced, okay? Usually they will just give you 2 marks, 2 marks. So sunlight is on the top, chlorophyll is on below. So it's chlorophyll, okay? It is not chloroplast because chlorophyll is a structure found inside chloroplast. So it's a structure responsible for trapping the sunlight. Yeah, so this is the chlorophyll, okay? Yeah, so just say about trap the light energy, okay? And then the light energy become chemical to produce a glucose later. Yeah, and then for this one, how are the carbohydrates made in photosynthesis being used and stored? Yeah. Okay, so just remember that. Okay, one thing is, whenever they perform the photosynthesis in the plant, right? Photosynthesis will produce glucose, but later, glucose will convert to become the sucrose. So for the glucose, they become the sucrose. Later, from the sucrose, right, they can become what? Starch. Yeah. So that's why glucose is being used by plant for several purposes, right? They can change to starch. So yeah. So why the glucose need to be, undergo the condensation, then become a starch, It's because glucose is soluble, but starch is insoluble. So this insoluble starch molecule, right, they are not able to react with the surrounding. So that's why they become an effective energy store. So that's why you need to remember that in the plant, right, they will store their excess form of energy in form of starch. But animal will store our excess energy, right, in form of glycogens. So that's difference. Okay, so that's why glucose is not being used to make the starch, right? Cellulose also, and also the glucose. Okay, glucose usually they are being used as a source of energy to use in the respiration. Okay, cellular respirations. Yeah, because they will undergo this one, which is this one, C6, HCl O6, plus O2, become what? Carbon dioxide. Okay, carbon dioxide and the water. So here's the equations, and then there is the energy. So here's the equation for the cellular aerobic restrictions, and then we require glucose as the substrate to start these reactions. So if we don't have the glucose, right, so that means the cell, they are unable to carry the cellular restrictions. The plant cell, if they don't have glucose, right, they are unable to carry out the cellular aerobic restrictions. So if they cannot carry out cellular aerobic restriction, right, they cannot release the energy to supply themselves. Okay, and then sucrose. Okay, sucrose is being used in the transport in the phylum, right, which is happen in the translocations. Okay, translocations. And then maybe as then they will ask you why plant prefer to transport in the sucrose form but not in the glucose form. It's because at sucrose they will not affect the water potential of plant. That's the reason. Okay, sucrose is very stable, not affect the water potential of plant. Yeah, and also glucose can be converted to another kind of compound. Okay, actually glucose can be converted become the protein fat also. This is not only occur in plant, this is also occur in the animal also. Yeah, we also can use the glucose, right, to build our proteins and fats. And then okay, you know about nitric iron already. Yeah, so that's why if you have not enough nitric iron, right, deficiency in nitric iron, what will happen to the plant? The plant will have a stunt growth. They cannot grow taller. Okay, they will have a yellowing of the leaf. And then for the magnesium, okay, magnesium are being used to make the chlorophyll. Yeah, so if you're lacking in magnesium, you have the yellowing between vein of the leaf. This is a bit different. Let me show you. Chlorosis is this one. Okay, chlorosis. Chlorosis is kind of the condition only happen in a plant. So which is they referring to? Starting from vein, so you can see the vein becoming yellowish vein. Okay, not whole leaf. It's a vein. It's a vein. Let me show you where is it. I put the picture at here. See, starting from the vein, leaf vein. So these conditions, now chlorosis. Because if the plant lacking of nitric ions, right, it refers to the whole leaf become yellow. Okay, so that's difference between them. Okay, like this. So what happens if the nitrate deficiency in plant? So you can see whole leaf just become yellow. Like this. See, this is totally become yellow. Okay, not only the vein. Okay, I think I put this picture. I think I can put this picture. So that's different. Okay, yellowing of the leaf. Let me adjust it. Okay, this also come out in the objective questions. Yeah, and then Adam Suyang, actually I'm still waiting for the homework. Okay, I think you have a lot of homework to complete, starting from I think chapter 17, right? Chapter 17, and then yeah. So it's okay. Later I'll list the homework down again, and then you can take your time to complete. Or if you need some time to complete the homework, we can delay the class first also can, and then at least you can have some time to complete the homework, and then we will have the class again. Okay, but it's all decided by you. Okay, it's all decided by you. Okay, this one. How does carbon dioxide get to the plant? Right, is enter, okay, carbon dioxide from the atmosphere, they just enter through the honey pot of leaf called stomata. Okay, so other thing you can just read by yourself. And then, okay, this one, limiting factor, limiting factor, okay? So here's the limiting factor. Light intensity, carbon dioxide concentrations, and the temperature. Usually it's this three. This three is the main factor. The volume of water and the concentration of chlorophyll, right, will be, yeah, it's also one of factor, but it is not so emphasized in the top. I mean in our syllabus. Our syllabus is actually focused on these three factor only. Okay, and then this one you can read by yourself. Yeah. Okay, they do some experiment. Okay, they do some experiment to study how the factors, okay, affect the photosynthesis. Okay, this one you can go through by yourself. Not really important. Okay, just need to remember, okay, like how the light intensity will affect the rates. So if you increase the light intensity, okay, so you increasing the light intensity, right, you are actually increase the rate of photosynthesis until they reach the plateau. The plateau. So whenever they reach a plateau, right? So if the question asks why, so you can say that because another factors, now they're becoming the new limiting factors. Okay, same to the temperature. Okay, but the temperature, okay, temperature is very important because temperature, right, cannot be too high. If the temperature become too high, it will denature the enzyme also, because photosynthesis process also a kind of enzyme catalyzed reactions. So that's why temperature cannot be too high. It should be optimum, okay, optimums, yeah, in the optimal range of temperature. For the carbon dioxide concentration is same to the light intensity, which is when you increase the carbon dioxide concentration, right, you will like reach a plateau again, okay, reach a plateau again until other factor become the new limiting factors. Okay, so here's a curve, okay, we need to recognize it, the curve. Okay, we continue to discuss. Okay, this one. Okay, this is very easy. Okay, I think I will not discuss again. Okay, this one, how starch is broken down in human digestive system. Okay, I think you should know how to do it. For this one, right, what's the marks? 6 marks. Okay, 6 marks. So that's why whenever you come with this kind of 6 mark question, you should be very, very detailed. Okay. So actually, digestion include 2 types of digestions: chemical digestion and the physical digestions. Yeah. So you should describe the process. Okay. That physical digestion is involved in the chewing actions and churning in the stomach. Yeah. Chemical digestion is involved in the enzyme, and then you should you can write down the keyword, which is from the large insoluble molecule becomes small and soluble molecule by the enzyme. Okay, so okay, we finished this already for this chapter. Yeah, and then okay, let me show you another question style. Okay, this one. Yeah, this is chapter 8. Chapter 8 starting from 2020. Okay, 2020. Okay, 2020. Yeah. And then you can read back. Okay, you can read back. I think that's all already. Okay, and then for this one, okay, this question is very interesting. Let me show these questions. 2022. Okay. Whenever you see this one, aphid insect. So this aphid insect, usually the question is related to the translocation, because aphid insect, they will just feed on the phylum sac. Okay. Why they feed on the phylum sac? Because phylum sac consists of the sucrose. They want to eat the sucrose. Okay. So yeah, so this is a way how you can prevent the aphid. You can kill them by insecticide or pesticide or any kind of chemicals. And then whenever the aphids feeding on the phylum tissue on the leaf, right? Yeah, state the name of the 2 tissue in the leaf. So that's why they actually penetrate from the upper epidermis to the xylem, to the pellucid vessel cell, to the spongy vessel cell. And then they reach the phylum. This is structure they penetrate inside. And then, okay, so they want to use this experiment to study the translocation of the sucrose in the phylum tissue. Okay, and then this is a glass chamber. So this glass chamber filled with the carbon dioxide gas containing this 40 isotope. Okay, this one is carbon label with 14 isotope. This is group A and group B, and then with the damp cotton wool. Okay, and then the leaf were enclosed in the airtight glass chamber. And then the special form of carbon dioxide gas, yeah, which is this one isotope, was supplied to leaf for a short period of time. So later, okay, the honeydew was being collected interval from the groups of aphids. And then the time taken for the sucrose containing the 14 carbons to travel the distance between A and B was recorded. Yeah, so they asked you to copy the table. Okay, so you have the 3 time of trials. So distance from A to B, right, is 650, 340, and 630. And then you record the time. And then you calculate the rate of movement of 14 carbon per hour. Okay, and then outline how the 14 carbon in the carbon dioxide gas become incorporated into the sucrose molecule and then translocated in the film. So where you can get the carbon dioxide? So that's why you get the carbon dioxide from the gas. Okay, I mean from the atmosphere. So that's why the carbon dioxide is a form of gas, right? Gas enter the leaf by what? By stomata. Okay, by the simple diffusions. So later the carbon dioxide will be uptake by the mesophyll cell. So it's being used in the photosynthesis. So that's why photosynthesis, okay, is a process involved in the chloroplast and being catalyzed by enzyme. So that's why this process, right, will produce a glucose. Glucose is C6H12O6. Glucose require carbons to become ingredients to build to build up the glucose, right? Which is the glucose later to convert it become the sucrose. So this is a process, okay? Because they just asking about how the carbon dioxide gas, right, can get into the sucrose molecule. So that's why from the carbon dioxide gas become glucose. So from glucose become sucrose. Transfer to all of plant. And then, okay, don't forget your sink and source. What is sink? What is source? I mean the source, okay? Sinks and the source. So the sinks are referred to the plant which they will store their nutrients. Okay. Sink for the sucrose. So the sink, right, including for what? This one. The growing seeds, flowers, or young growing leaves. They are example of sink. Okay, and also the buds or root tips or shoot tip. Yeah. You just pick two answers. Enough for you already. Okay, I just state all of the possibility. Okay, and then this one use of sucrose by the sink. How you're going to describe sucrose by the sink is that once the sucrose, right, they reach their destinations, and then they will convert it back, become glucose. Means that when the sucrose being translocated within the film, right, and then whenever any kind of plant cell they require to do the respirations, so the sucrose will be converted back to glucose because plant cell only can uptake the glucose to do the cellular aerobic respirations. So that's why glucose is being used for the cellular aerobic respiration to produce energy. Yeah, and also the energy is being used for what? For the growth, energy transport, and the cell divisions. Okay, so those processes, right, require energy. And also when the plant they grow new leaf or new fruits, right, they also require energy input. So those energy input are get from the glucose. Okay, so other kind of usage, you can say that sucrose becomes starch, a source for cell wall. Yeah, so this is the question. Okay, use of the sucrose by the sink. And also glucose can be used to convert it into the amino acid to build the proteins. So you can see it's always repeating. And then this one, okay, this one you can read by yourself. Positive phototropism, and then this one. So that's why, and then they like to ask you about how you can identify where is xylem, where is phloem. Okay, I hope you know. So you see here is xylem, here's xylem. The big hole, okay, here's it, the big hole, xylem. Okay, phloem tissue. Okay, whenever you talk about phloem tissue functions, you need to write the keyword translocations, and then what kind of nutrient molecule? Glucose, amino acid, and then from where to where, source to sink. This is also same to the xylem. Okay, so you can read about this one. Okay, I have do the analysis for the past year. Okay, this is chapter 8. Okay, I just talk about chapter 8 first. chapter 8, okay, you can have a read later. Only about 4 or 5 pages, okay? Because I hope that this document can help you to focus on the right thing. Because I worry that you feel like a lot of things to memorize, right? You don't know where to focus. So you can read this first, and then you should know how to focus. Yeah, so you can see, right, the trends of the question, right? Now they are moving to this one, applications. Okay. Especially asking you this one, hydrophyte and xerophyte. So that's why hydrophyte and xerophyte is a must know. It's a must know questions. Okay, you need to know about the detail for the hydrophyte and xerophyte, their adaptations. Okay, and then the source and sink. Yeah, so this one you can read by yourself. And then describe how xylem is adapted for functions. Okay, so this feature. Okay. Yeah, so this is the answer you can write. Okay. Okay, I should add this from source to sink now. Okay, forget to write this. Yeah, from source to sink, and also transfer water mineral ion from where? From the roots, right, to all part of the from the roots to leaf, right? To leaf. And then xylem adaptations. Okay, should write this. Yeah, there's that cell with no cytoplasm inside, and then please add on the reasons. Okay, because they usually ask you to explain, right? Explain. So that's why they can allow continuous column of water. And then the leading, right, they can provide supports to the plants. No crossroad, right? They can form a continuous column of the water. Yeah. And then for the water uptake by the root hair cell, so that's why the root hair cell, right, this is their adaptations. Yeah, they are very long, increased surface area, thin cell wall, a lot of mitochondria. And then active transport, okay, you should know the point. And then transport mechanism, right? Referring to the how plant uptake the water and ions, okay. And then transpirations, right? Transpirations. Okay, you should know how to describe the process in the transpiration, which is starting from the evaporation. See, first, water evaporate from the surface of nucleus cell. The keyword, okay, write down the keyword evaporate from the surface of nucleus cell into the airspace. Later, water diffuse out to the atmosphere through somata. The keyword diffuse out through the somata. So this will create the tension on top of plant. So later water are held together by cohesions. Yeah, cohesions. So they become a continuous column up. Continuous column of water up the xylem. So later the water will enter the root hair cell to replace they want to replace water loss, okay? So by osmosis. So actually you have two types of force. One called cohesions. Cohesion is referred to sticking between the water molecule, and then this one adhesions, right? Adhesions between the water molecule stick to the wall. Okay, the water molecule, right? Yeah, sticking to the wall of the xylem vessel. Okay, and then this factor, okay, just read by yourself. Okay, take note of this humidity. If you have high humidity, you have a lower rate of the transpirations. Okay. And then translocations, right? Translocations. Describe them. Okay, describe them. Yeah. Yeah, the source and the sink. Okay, you should know how to recognize it. Usually the source is referring to the leaf, and then sink is referred to other kind of organs. But sometimes due to the seasonal change, right? The part, right, which is initially is a sink. Okay, is a sink. Now they'll become the source. For example, in the potato, the tuber, now they become the source. Because they can break down the starch, become the sucrose. Usually they store their excess energy in form of starch, right? So whenever in the winter time, the plant are able to do the photosynthesis. So they still need to they still need to survive, right? So that's why now the leaf, right, they are not becoming the source because leaf they cannot do the photosynthesis in the winter season. So now the tuber become a new source because inside the tuber, right, they store a lot of food inside. So they will break down the starch inside to become the sucrose and help in the translocations. So that's why the tuber become the source, and then the growing shoot become the sink. So that's why the tuber, right, they will provide the nutrient, which is the sucrose. So later they will translocate to the sink. Okay, so later the sucrose will convert become the glucose. So glucose can be used for the energy for growth. Okay, so this is in the spring, okay, in the spring or in the winter, which is the leaf cannot do the photosynthesis. But in the summer time, which is they have a very high light intensity. So the leaf, they are source of photosynthesis. Yeah, so the new tuber are the sink because it's the one who store the nutrients. So take note of this one, a bit tricky. Okay, so remember about xerophyte and the hydrophyte. Okay, which is their keyword to write, okay, their keyword to write. And then this also, yeah. Okay, some of mistake to avoid, which is if some of the I mean a lot of students, right, they will write in this way: water evaporate from stomata. Okay, so it's not evaporate from stomata, it's diffused from stomata. Water will evaporate from the mesophyll surface. Later they will diffuse through the stomata. Okay, please don't do your mistake. Water diffuse from the stomata. Diffusion, okay, is by diffusion because oh, okay, maybe I can add something. When the water evaporates from the mesophyll surface, right, the water, after they undergo evaporations, they will become water vapor to the bit. Water vapor is a gas, it's a gas form. So that's why later inside the airspace, right? Okay, inside the airspace, this is in the airspace, okay, in the airspace. So later the water vapors will diffuse through the stomata to outside. So just remember that phloem flow in both ways. Xylem only goes up, okay? Only go in the upward directions. Okay, that's all. This is chapter 8. Okay, now we talk about chapter 6. Give me just 4 minutes. Just few minutes only. Okay, chapter 6. The thing you need to know is also like focus on these two kind of plant. And then okay, you should know how to write the equations, okay? And then what is the usage of glucose? A very common question, okay? Usage of glucose. And then leaf structures and adaptations, right? Okay, so that's why usually the leaf structure, they will ask more about the hydrophyte and xerophyte in this lesson 2 year. Yeah. So that's why you should recognize the positions, okay, for the hydrophyte and xerophyte. And then each of the function, okay, and their feature. So just remember that stomata, they can allow diffusion of gas and also water vapor loss. Okay, and then this one, okay, which is the same thing that I discussed with you just now. Yeah. So this is some of the questions they ask you, okay, which is they ask you how you're able to describe stomata distribution in the hydrophyte. So you can say that stomata only located on the upper epidermis. Yeah, why they do thin cuticle? Because they don't need to prevent water loss. And then the xerophyte, right? They'll give you this one, marram grass cross sections, okay? And then describe the feature, yeah. And then label it, label it. Okay, and the limiting factors, right? Limiting factor, okay, which is they focus on these three one, and then they will give you a graph. So you need to understand the trend of the graph, right? So that's why you need to describe the graph trends. Yeah. So which is the rising part. The factors is become limiting factors. So at it increase, right, rate also increase. And then when they reach a plateau stage, right, the factors on the x-axis become no longer limiting. So they will become other kind of factors. Okay, like this. Yeah. And then interpret the graph. Okay, which is the same thing now. And then the glass half. Okay. Discuss the advantage of the growing crop in the glass house. So the advantage is what? Is because you can control the factors, which is the light, temperature, and carbon dioxide in the very optimal conditions. So that's why you can have a higher rate of photosynthesis, and then you can higher yield of the productions. Okay. Yeah, and then the function for magnesium and nitrates. Yeah, this one. And then for this one, okay, you need to know how is the change in color for the hydrogen carbonate indicator. Yeah, this one. Okay. You know about this one. So just remember that plant is in the light condition, right? When it's the light conditions, they refer to the low carbon dioxide because photosynthesis rate is higher than the respiration rate. But when the plant in the dark state, right, respiration rate is higher than photosynthesis rate. Yeah, this one. Okay, this is read by yourself. Okay, that's all. That's all for chapter 6 and chapter 8. Yeah, so we already finished revisions about the chapter 6 and chapter 8, which is a plant. Plant. Okay, and then you can read back. Okay, read back this question. Okay, yeah, you can read those past year question. Okay, past year question and the answer, and then I'll give you some questions to try. Okay, just a few questions only, and then revise on this one also. Okay, so you need to read on the hydrophyte and xerophyte. Okay, I'm not sure you will come out in this year or not, but last year they do come out few times already for the hydrophyte and xerophyte. Okay, so that's all for today. And then I'm waiting for the homework. Okay, so you spend some time, okay, to do the homework for the paper 4 and also paper 6. Okay, I'm still waiting for the paper 6. Okay, that's all. Bye. Bye. Okay.